Hello. Please hit like button and subscribe my channel. Also press bell icon for future video notifications. Thanks.
Researchers found that two toxins produced by the Vibrio genus of bacteria cause actins to start joining together into these filaments, which could be thought of as cellular highways on which cargo is delivered, at the wrong location inside cells, and headed in the wrong direction. Growing in the wrong direction is a totally new function that was not previously known and was not thought to be possible for actin filaments inside the cell, said senior author Dmitry Kudrashov, Associate Professor of Chemistry and Biochemistry at The Ohio State University. A large fraction of actin in the cell is consumed in formation of the highways, where they are not needed, so the cell resources are wasted and cannot be used to satisfy the cell's basic needs. The research is published today, November 18, 2022, in the journal Science Advances. In this study, the research team zeroed in on describing the unexpected cellular activities rather than any further implications, such as how the hijacking relates to bacterial infection. We are looking at the interference at the molecular level. We have not focused here on how this cell function might affect humans, said first and co-corresponding author Elena Kudrashova, a research scientist in chemistry and biochemistry at Ohio State. But finding something that we didn't know was possible, for actin to behave in such a way inside the cell, raises new questions about whether this function might actually be needed, or could come about in some other way. Until now, actins have been known to assemble each filament in one way, or because they are limited in number, the actins disassemble as needed from the pointed end and are recycled to maintain directional activity toward the barbed end, and then these actin filaments perform functions, such as cell migration, contraction or division, as dictated by what the cell commands. The toxins start making these actin filament highways in the wrong place, building something that is useless for the cell, and the cell doesn't know how to deal with it, Kudrashov said. Though they don't yet know all the consequences of this hijacking activity, the researchers said the results could include seepage of nutrients through damaged intestinal walls, which would provide food for the infectious bacteria waiting outside. And that's why the scientists want to learn more, whether other molecules can force actins to assemble roads to nowhere, and whether that strange filament formation might even be a beneficial mechanism under a different set of circumstances. It's quite possible that our own cells are doing this on some occasion, but we don't know because actin has so many functions and not all of them are yet well understood, Kudrashov said. The Ohio State team collaborated with co-authors Ankita, Heidi Ulrichs and Shashank Shakar of Emory University. This work was supported by grants from the National Institutes of Health. Materials provided by Ohio State University. Original written by Emily Caldwell. Note: content may be edited for style and length. Elena Kudrashova, Ankita, Heidi Ulrichs, Shashank Shakar, Dmitry S. Kudrashov. Ohio State University. 2022, November 18. Toxins force construction of roads to nowhere. Discovery is first report of effect on the abundant actin protein. https://ift. tt. htdxrfc. Accessed November 18, 2022. Thanks for watching. Please subscribe my channel for more videos.